Hello, and welcome to Lupus and CAR T Cells. I'm Annika Hazlitt, a member of the Board of Directors of Kaleidoscope Fighting Lupus, and your host. CAR T cells have the potential to revolutionize lupus treatments. They may even bring about remission for many living with lupus. Here to explain the science behind CAR T cells is Dr. Don Thomas, a clinical rheumatologist and the author of the Lupus Encyclopedia. Before we hear from Dr. Thomas, I'd like to thank our sponsors, Tin Rocket, Blue Eyed Crow Studio, 7 Minute Cinemas, and Pharma for their generous support in making this educational presentation possible. Now, let's hear from Dr. Thomas.
Well, thank you very much for that kind introduction. Today I'm really excited to be here to talk about an exciting new therapy for lupus called CAR T cell therapy. Is this a new chapter in the treatment of lupus? I certainly hope so. So first, let's go back to April 2021. There was this young woman in Germany. She was Asian descent, just 20 years old, but she unfortunately had severe systemic lupus erythematosus. It was attacking her heart, like the valves of her heart, the lining of her heart called the pericardium. She had severe lupus nephritis where it was attacking the kidneys. She had horrible arthritis. She had rashes from her lupus as well. And she was dumping over 2 grams of protein in her urine per day. We call this proteinuria, and that's because of the lupus nephritis. Normally, you should be dumping less than 200 milligrams of protein of urine per day into the urine, and she was dumping over 2,000. That's how bad her lupus nephritis was. Her C3 complement level was low. Her anti-double-stranded DNA level was high, and these can be signs of active systemic lupus. And she had unfortunately failed all therapies. She had failed cyclophosphamide, mycophenolate, steroids, and several others. Where her doctors, they were trying to figure out what they could do next to help her out. And Dr. George Schett and his team, who were her German doctors, they had read about this therapy that had been given to mice with lupus that put them into complete remission called CAR T cell therapy, so they decided to give her that. Now, in order to measure how severe her lupus was, they measured something called the SLEDAI score. SLEDAI stands for Systemic Lupus Erythematosus Disease Activity Index, and it's a way we can measure how severe someone's lupus is at the beginning of a research study and then at the end, and it can tell us if the treatment works or not. Her number of points was 16. All you need is over 10 to be considered as severe systemic lupus erythematosus. So she really had severe lupus. So she was given the CD19 CAR T cell therapy. And by the way, the reason it was available is because we use it to treat patients who have B cell cancers, like B cell leukemias and B cell lymphomas. They started to become available in 2016. Well, 1 month after treatment, she was completely in remission. Her SLEDAI score was down to 0. I have never seen a patient treated with that severe of lupus and have complete remission within a month. That's just miraculous. Well, Dr. George Schett, he showed a photograph of this young woman after her treatment. And the photograph looks very similar to this. This is a different photograph I got off the internet, but she just looked so happy. And it was a drug-free remission, by the way. She was on no treatments whatsoever for her lupus. The reason she was going in to see the doctors was for them to see how her lupus was doing, but not to monitor any drugs because she wasn't even on any drugs. So they went ahead and they've been using it in other patients, and in September 22, they published a report of their first 5 patients treated with CAR T cell therapy. All of them went into complete remission. And that's when social media started to really light up about CAR T cell therapy and lupus patients, possibly curing lupus patients. And that's why we're here today to talk about CAR T cell therapy. In November 2023, we had our American College of Rheumatology yearly scientific meeting. And at that meeting, so far, we have 24 patients with severe systemic lupus who have been treated with CAR T cell therapy. 22 of them are in complete drug-free remission. And 2 of the patients were close to remission, but not totally in remission because it was just too soon to tell. So again, this is just very exciting. But what is CAR T cell therapy? I'm going to try to simplify it to make it easy to understand. Well, CAR T stands for chimeric antigen receptor T cell. So there's 3 different parts to CAR T. First, this is an image of what it looks like, and I'll go over the different parts of the CAR T cell in the next few slides. So first, it is a T cell. T cells are a type of white blood cell. I already mentioned B cells. B cells are the types of white blood cells that make antibodies like anti-nuclear antibody, anti-double-stranded DNA antibody. Well, T cells are a different type of white blood cell. It can tell B cells to become more active. It can tell B cells to be less active. It can also destroy cells as well. So they take T cells from a patient and then they attach an antigen receptor on the surface of those T cells. And I'll explain what an antigen is. An antigen is a molecule that can be recognized by an antibody, and they fit together like a lock and key. Anti-nuclear antibody is an antibody, anti-double-stranded DNA is an antibody that the lupus immune system makes, and then the antigens would be the nuclear proteins or the DNA. Well, this antigen receptor on the surface of these T cells, it can recognize other molecules or antigens that are on other types of cells, and we'll talk about that in a lot more detail. In addition, it's chimeric. So what does chimeric mean? It refers to a Greek monster called the chimera. The chimera he had the body of a goat, the head of a lion, and the tail of a snake. So it's an organism that is made up of cells of more than one organism. So that's a way to think about it. So this CAR T, chimeric antigen receptor T cell, the T cell actually comes from the patient. We take the T cells out of the patient with systemic lupus. And then we insert DNA into the nucleus of these T cells. So this is the different organism that's being put into the patient's T cells. And then that DNA causes the T cell to produce this antigen receptor that's on the surface of the T cell. And so it's chimeric because it's made of different parts other than just the patient's own T cells. Now, what is the actual process? If I tell you that I want to treat you with CAR T cell therapy, what would you go through? Well, the first thing we would do is we would taper you off of your immunosuppressants. So we would get you off your mycophenolate, your methotrexate, your azathioprine, at least within 3 weeks before therapy. In addition, we want to decrease your prednisone to less than 10 milligrams a day or less because we don't want it to interfere with the therapy. Then we would have you come into the clinic and we would take blood out of you about 13 days before your CAR T cell infusion. The reason for that is we want to take your T cells out. We want to separate them from your blood, and we do this by using a machine called an apheresis machine. Apheresis means taking blood out of a patient. Now, in this particular case, we're doing something called leukapheresis. Leukapheresis means we're taking the white blood cells out of the patient. Leuko comes from the Greek word for white. And on this apheresis machine, you can see the yellow bag hanging up on the left side. That contains white blood cells, while the red ones next to it are the red blood cells, and those are infused back into the patient after the apheresis treatment or the leukapheresis treatment. Then the next step is the T cells are taken into the laboratory and they're mixed with a virus called lentivirus. And I'll explain that in more detail. Now, lentiviruses actually exist in nature. And most of you have heard about one of the most common ones, which is the HIV virus. HIV is the virus that causes AIDS, and it is a lentivirus. So the way that HIV happens is that when HIV infects a person, it can recognize T cells and it attaches to T cells, and then it infects its DNA into the T cell and makes the T cell replicate and multiply more of the HIV virus. And then the HIV virus destroys the T cells. They go throughout the body to infect more T cells, and they keep reproducing themselves over and over. So the doctors and the scientists use the lentivirus because it's able to attach and recognize the T cells, but they trick it. They take out its own DNA and they put in a type of DNA that is programmed to make whatever we want it to make. And in this case, an antigen receptor that can attach to B cells and destroy B cells. And so the T cells that are taken from the patient are mixed together with this lentivirus. The lentivirus attaches to the T cell, and then it infects the T cell with the DNA that's inside of it. Then the DNA is put into the nucleus, and then the DNA makes the T cell start to produce antigen receptors, whatever type we want it to make, on its cell surface, and voila, the patient's T cell has become a CAR T cell. And then these CAR T cells are replicated and multiplied over and over again inside of the lab. So now what's the next step? Well, we bring you back into the clinic 5 days before your CAR T cell infusion. The CAR T cells, by the way, are sitting in the laboratory, and the technicians are having them replicate and multiply and increase in numbers. But we bring you back into the clinic and we give you a low dose of chemotherapy. We give you one called fludarabine 5 days before your CAR T cell infusion. Then we give it 3 days in a row. And we give you 1 dose of cyclophosphamide 3 days before your CAR T cell therapy. And the reason for this is we want to decrease the number of T cells in your body. So this chemotherapy decreases the T cells significantly. And we want that to happen because we want the CAR T cells to expand inside of your body and take the place of those T cells that are missing. So then we give you back the CAR T cells, we infuse them back into your body on the day of your infusion. The CAR T cells go into your body, they notice that there's not very many T cells around, and that causes them to start to replicate and multiply. And they expand and replicate within the body. But the other thing they do is that they can identify and attach to B cells, and that signals them to multiply and replicate as well. So this particular CAR T cell that was used by the German doctors, it was engineered so that the end of the antigen receptor could recognize a particular antigen called CD19. CD19 is a molecule that's only found on some B cells. It's not on all B cells, but it's on some B cells. And notice that the CD19 in this picture is kind of triangular in shape. And then the antigen receptor is the same shape, and they come together like a lock and key. And that's how antigens and antibodies work, or antigens and antigen receptor in this particular case. So they come together, they lock together like a lock and key, and this signals the T cell on the inside of the antigen receptor to tell this T cell to destroy and kill that B cell. But not only that, what it also does is that the signaling tells this particular CAR T cell to multiply, because it thinks these B cells are foreign invaders and they want to kill all the B cells that they can. So the CAR T cells start to replicate and multiply because of this activation that occurs from the antigen receptor. Then the CAR T cells just start to destroy all these B cells throughout the entire body. In fact, by 2 to 7 days after the infusions, these 5 patients, all of their B cells were completely depleted and gone within their body. Now, why should we get rid of B cells in lupus patients in the first place? And that's important to know to understand why this type of CAR T cell therapy can help. Well, it's helpful to know how the normal immune system works first. So our immune system is there to protect us from foreign invaders. And let's just take the example of bacteria. On the top of this slide is a type of bacteria called streptococcus that can cause strep throat. The bottom of the slide is another type of bacteria called staphylococcus. And surrounding these bacteria are all kinds of molecules and proteins that are different from the other bacteria. And we call all of those proteins and molecules antigens. And there's a lot of different ones on both of these bacteria. Well, the type of blood cell inside of our body that can recognize those antigens is a type of white blood cell called B cells. B cells are also called B lymphocytes. One B cell can only make one type of antibody, and that particular antibody only recognizes one type of antigen. And in this particular case, you notice that the round antigens of the streptococcus fit perfectly with the antibodies being produced by that B cell on the left. So when the person gets infected with strep throat, if that particular B cell comes into contact with the bacteria, it tells the B cell to become active and to attack the bacteria and destroy it. The same thing with the one on the bottom. The B cell on the bottom, notice how it's square in shape. It's not round, and it can recognize the square-shaped antigens that are on the Staphylococcus bacteria, and it can recognize no other antigen within the body, but it can attack and destroy the Staphylococcus. And here's an example of Streptococcus being attacked and swallowed up by a white blood cell. So what do we see in the body itself? Let's say that someone does get infected with Streptococcus. The streptococcus goes through the throat and infects it, but the B cells that can recognize it, like I showed on the previous slide, notice that it's there, and then they start to replicate and multiply, and then not only that, but they also start to have other white blood cells come to the throat to the rescue to attack and destroy the bacteria. Then you start to get swollen glands, and then we look down into the throat, and this is what we see as the physician. Notice the white areas. Those are pockets of purulence or pus on the tonsils. Those are actually collections of those white blood cells. Well, the immune system is amazing that it can protect us from these bad foreign invaders, but a downside is that it can cause inflammation, and inflammation can be harmful. And we can clearly see the signs and symptoms of inflammation on this photograph. Inflammation can cause swelling. You can see the swollen tonsils. Redness, you can see the redness that's going on. Pain, if anybody's ever had strep throat, you know how painful it can be. It can be so painful it's hard to even swallow water. And then fever can occur, and then also fatigue as well because of the systemic inflammation that's going on. But doesn't this all sound familiar? These are also things that happen in lupus patients. You have B cells that are making antibodies that think your own tissues and cells don't belong there, are attacking them, and then cause inflammation like this, and that can actually destroy organs of your body. And so we can get rid of those B cells and try to help you out. So what happens in the lupus immune system? Well, in the lupus immune system, lupus patients have a type of B cell that are bad for them. We call them autoreactive B cells. Auto means self, and these are B cells that actually make antibodies that think that cells that belong in the body itself are foreign, and they think that they're foreign invaders and can attack those particular cells and tissues. And for example, in this picture of this man who has systemic lupus and he has the classic malar rash on his cheeks, it thinks the skin doesn't belong there, and so it has antibodies that are attacking the skin, and the white blood cells are attacking the skin, causing the inflammation. And you can clearly see the inflammation with the redness. If you feel it, it's a little swollen, might be a little bit tender, might be warm to the touch as well. So inside of our body, we have trillions of different cells. These cells, they also have nucleus inside of them. Inside of the nucleus, there's also lots of other proteins like DNA. And then just think about it. Almost every lupus patient watching this video has anti-nuclear antibodies. So you have B cells, bad B cells, that are making antibodies who think that the proteins inside of your own nucleus do not belong there. 70% of you have anti-double-stranded DNA antibodies, and these think that the DNA inside of your own cells don't belong there. And then all of this can end up causing your lupus inflammation. So just think about it. What if we could get rid of the bad B cells, and then this could result in getting rid of those bad autoantibodies that attack the person? Could we help lupus patients? And the answer is yes. We're already using medicines that decrease B cells. There's Benlysta, which is belimumab, Rituxan, which is rituximab, and Gazyva, which is obinutuzumab. All of these decrease B cell activity in lupus patients and are very helpful. But these medicines don't work in everybody. And there's some reasons for that. Number one, these medications, they only work in the blood and the lymphatic system. Lymph is a type of fluid that flows through our body outside of the blood system of our body. So these medicines, they don't get rid of or deplete the B cells that are hiding in the tissues. So the person with lupus arthritis, it still will have bad autoreactive B cells inside of the tissues of the joint that are not being depleted by the Benlysta or the Rituxan. And so because of this, if you were to stop the medicine, the B cells in the bloodstream might be much lower, but they'll start to come back because there's these autoreactive B cells or bad B cells that are still in the tissues, and they can just start making more of those bad B cells. So you have to keep giving Benlysta and Rituxan over and over again in lupus patients. So now let's talk about 8 patients that have been treated by the German group. I showed you that first patient who is 20 years old and Asian ancestry. She was the very first patient ever treated by the German group. Well, in November 2023, this German group presented the results in 8 patients it had treated with CAR T-cell therapy that was specifically targeting CD19. And we'll talk more about CD19 in just a few slides. And how well did they do? Well, they can measure some things in the patients. One of those things was anti-double-stranded DNA levels. And on the left side of this graph, you can see the levels of all 8 of the patients. And it shows the level of the DNA. A normal anti-double-stranded DNA level should be less than 40. There was only one patient who had a normal anti-double-stranded DNA level. All the other ones were very high. One of them was close to 6,000. The higher the anti-double-stranded DNA level, it oftentimes correlates with the severity of their lupus. And when we treat these patients with good medicines, we usually see the anti-double-stranded DNA level come down. Doesn't happen in everyone, by the way. Some of you have anti-double-stranded DNA levels that stay the same even if you go into remission. But in these particular patients, this did correlate with their disease activity. Then they followed them every 3 months after that. Look what happened at the 3-month mark. Every single patient, their anti-double-stranded DNA level went to 0. I have never seen a therapy for lupus where every patient, their anti-double-stranded DNA level goes to 0, especially at 3 months. And then they followed them every 3 months after that, and every 3 months after that, they still had no anti-double-stranded DNA. Well, blood results are good, and it's a great way to be able to follow how good our lupus patients are doing, but it doesn't always tell us how the lupus is doing overall. So another thing they can measure is the SLEDAI score, like I mentioned earlier. And this graph here, it shows the SLEDAI scores of all the lupus patients. Remember I said that above 10 is severe systemic lupus, and you can see that all of these patients had either severe systemic lupus or very close to severe systemic lupus when they first got their CAR T-cell therapy. So what happened at the 3-month mark? At 3 months, every single one of them was in 100% remission except for one, and that particular patient went into remission very soon after the 3-month mark. Then at every 3 months afterwards, those patients continued to be in remission. And not only in remission, they were in drug-free remission. They were not taking any immunosuppressants whatsoever. Some other interesting things happened. Number one, the patients who had positive anti-Smith antibody, anti-Smith is an antibody that's very specific for lupus, and we almost rarely see someone who doesn't have lupus that's anti-Smith positive. Their anti-Smith antibodies went to 0. I have never seen anti-Smith go to 0 in any of my patients over the 30 years that I've been treating lupus patients. So this is phenomenal. It tells us that it's getting rid of the bad B cells that make anti-Smith antibody. In addition, interferon type 1 normalized in all the patients, and lupus nephritis went away in every single patient as well. Interferon type 1 is a type of cytokine. It's high in 80% of our lupus patients, and it correlates with disease activity. You might have heard of the medication called Safnello. It's also called anifrolumab. It actually works by decreasing interferon type 1 levels. Well, that's what the CAR T cell therapy did as well. Now, B cells, not only can they make the bad autoantibodies, but they're very important for doing other things like protecting us from infections. And so one of the fears is with getting rid of all these B cells, we be reducing the responses to vaccines in patients? Well, the good news is that all of these patients, after their CAR T cell therapy, were still producing antibodies against those bacteria and viruses that they were vaccinated against. They were still active vaccine antibodies against measles, mumps, rubella, also tetanus. Their COVID vaccines were still working, and their pneumococcal vaccines were also still working. A few of the patients got a new vaccine after their treatment with CAR T cell therapy, and they had a good response. The reason for this is because I mentioned earlier that CD19 is located on some B cells, but not all B cells. There's a type of B cell called plasma cells that do not have CD19, and they were still able to make all these antibodies against the vaccinations. So what happened to the B cells in these 8 patients? Well, in all 8 of them, all of their B cells went completely away. And then around day 100, they started to come back on average. Some of them was as early as 90 days. Some of them was as late as 115 days. Well, the doctors were really nervous when the B cells started to come back, because the question is, could these be bad B cells? Could they be the B cells that are from the lupus patients? Well, guess what? These B cells were actually normal B cells. And how in the heck does that happen? How can it happen since we're destroying all the B cells in the blood and the lymphatic system? The reason is because the most immature B cells don't have CD19. And so those premature B cells that are in the bone marrow could still make normal B cells. So the scientists and the doctors in the research studies stated that it seems like CAR T cell therapy resets the immune system in lupus patients. It comes close to normalizing the immune system in lupus patients, and that's really exciting. However, I do disagree with that because it doesn't reset the immune system in everyone. One of the patients was anti-SSA positive. Some lupus patients are positive for anti-SSA. It comes from the word Sjogren's syndrome A, and that's because 2/3 of Sjogren's syndrome patients are positive for anti-SSA antibody. Some lupus patients are as well. But this one patient who was anti-SSA positive, the anti-SSA antibodies never went away. So that tells me that it still had the B cells, those autoreactive B cells that produce anti-SSA antibodies. So it's not normalizing the immune system or resetting the immune system in every lupus patient. So is CD19 CAR T cell therapy right for Sjogren's disease patients? Is it right for those lupus patients who have anti-SSA antibodies? And it's a big question mark. We really don't know, but we have to wonder if that could possibly be the case. Another potential problem is that the immune system in every lupus patient is different. For example, there's over 180 different genes that cause lupus to occur. And those genes, they cause different abnormalities of the immune system. Even though they might cause similar problems like the malar rash and lupus nephritis, there's different parts of the immune system that are being affected. Some patients, the interferon might be the most important part. Others, it might be the T cells. Others, it might be a type of white blood cell called macrophages. Should we be making CAR T cells that actually target those particular cells and those particular lupus patients? We need to figure that out, and there's a lot of studies that are being done right now to see if we can answer that particular question. It's helpful to know how B cells mature in the body, and so this is a nice image that shows that. On the left side of the screen, you can see pre-B cells, which are a very premature type of B cell, that when it becomes activated, it becomes an immature B cell. Then that, if it's a healthy active B cell that's not going to hurt the body. It goes out into the bloodstream, and then it matures all the way up to a plasma cell like you see on the right side of the screen there. One of the last most mature B cells is called a memory B cell. Memory B cells, they're there and they can recognize one particular antigen, but they're not making antibodies yet. But they're there at the ready. They think of them as soldiers that are on the lookout for foreign invaders. So if a memory B cell can recognize the pneumococcal bacteria, for example, or the streptococcus bacteria, then it can become active, start to make a lot of antibodies and start to multiply and then mature into the plasma cell, which is the most mature type of B cell. Plasma cells are constantly making antibodies. In fact, they make about 2,000 antibodies per second. Just think about that. In 10 seconds, one plasma cell will produce 20,000 antibody particles. And then there's millions of these plasma cells throughout our entire body. And some of these plasma cells, by the way, can be immortal as well in making antibodies throughout our entire lifetime. Now, all these different B cells, they have different molecules or antigens on their cell surfaces. And you can think of these antigens as being badges. And some of them have some antigens and not others. For example, there's one called the CD19, which is the CAR T cell therapy. Recognized this particular antigen in the German study, for example. CD19 is present on the premature B cells as well as all the way up to the memory cells, but it's not located on plasma cells. And that's why the vaccine still worked in those patients. Another example is one called CD10. That's an antigen that's only found on the most immature B cells. And then there's another one called CD20. CD20 can be found on more mature B cells, but not on the plasma cells, nor is it found on the pre-B cells. And then there's another antigen called BCMA, which is only found on plasma cells and plasmablasts, which are immature plasma cells. The bottom line is that these antigens, they help us recognize and distinguish one type of B cell from another. And each stage of the B cells has different types of antigens. And you can think of it this way. Let's say that you have 10 people standing in front of you. All those 10 people have a nose and ears and eyes and cheeks and hair. But each of those has a different size and a different color, and that particular combination helps you recognize, yep, that's my brother Tim, or oh, that's my sister Michelle. Same thing with these B cells. We can recognize the exact B cells by their particular antigen patterns that they have. So the CAR T cell therapy for CD19, it attacked these particular B cells, leaving the plasma cells alone. There's actually medicines out there for CD20, Rituxan, which is rituximab, and Gazyva, which is obinutuzumab. They destroy the B cells that contain the CD20 on their cell surfaces. And there's the BCMA that I mentioned. BCMA stands for B cell maturation antigen. It's not on those other B cells, but it is on the plasma cells and on plasmablasts. And by the way, this was actually used in the Chinese group. There was a Chinese study using CAR T cell therapy in lupus patients, and they actually presented their data on 13 patients. And they've actually been studying this CAR T cell therapy even before the German group. They just didn't report their results before the German group. But they used an interesting type of CAR T that recognizes both CD19 and BCMA. So it destroys all of these B cells that you see on the screen. So which type of B cell antigen is the best one to target for lupus patients? Is it CD20? Is it CD19 like in the German group? Is it BCMA? There's one that's being studied right now in lupus patients called Descartes-08 that only targets BCMA. Or is the Chinese group one the correct one to do that recognizes and destroys both BCMA and CD19? So let's go back to the B cell drugs that I mentioned earlier. Why would CAR T cell therapy work better? Because we're getting 100% remission rate so far in these lupus patients, but when I give someone Benlysta or Rituxan, it doesn't work in a lot of patients, and we have to keep giving the medicine over and over. Well, like I mentioned earlier, Benlysta and Rituxan and Gazyva, they deplete the B cells in the bloodstream and in the lymphatic stream, but not deeply inside of the tissues. And that's the advantage of CAR T cell therapy, which can deplete all of the B cells, even within the tissues, because the CAR T cells go deep into the tissues to destroy them. But we don't see that with the Rituxan or with the Benlysta. So far, there's 24 lupus patients who have been treated with CAR T cell therapy. And this is as of November 2023 at our American College of Rheumatology meeting. It was several different studies, and I'm putting all of them together. But out of all 24 patients, all 24 had severe systemic lupus, all 24 had failed multiple therapies. 22 were in complete drug-free remission. 2 of them were very close to remission, and the only reason they weren't in complete remission yet is because it was just too early. One of them had only been 1 month since therapy, one was only 2 months since therapy, but I wouldn't be surprised if they're in remission at the time that I'm doing this video right here. So how about side effects? You know, it just sounds too good to be true, doesn't it? We're seeing these miraculous cures or remissions, and none of them had any severe side effects. But we do have to worry about it. We're getting rid of B cells from day 9 to day 90, and B cells are important for fighting off infections. So we have to worry about the possibility of infections. Well, in all 24 patients that were reported, there was only one severe urinary tract infection that occurred, and that was in one of the Chinese patients, but the patient responded very well to therapy. Now, I will have to mention that someone reached out to me on social media and told me that one of the 24 patients ended up getting a severe infection that put her into the intensive care unit. She did well and came out, but that could be a possibility. This was actually a doctor who sent me this message, but you can't always believe everything on social media, and we just have to wait to see what the next reports in the medical literature actually show us happen to the patients, but we always have to keep in mind the possibility of infections. Another potential complication of CAR T cell therapy is something called cytokine reactivation syndrome. This is similar to something called cytokine storm. During COVID-19, many of you have probably heard about cytokine storm. It was actually a major cause of death in many people, especially during the first year of the pandemic. What happens is that cytokines are molecules that tell white blood cells what to do. So that's how white blood cells can talk to each other. Some of them increase inflammation, some of them decrease inflammation. But these actually, with the cytokine storm, they increase inflammation. When someone gets infected with COVID, the immune system can become overactive and just go crazy, start to make all kinds of cytokines, and then attack the organs of the person and kill the person. Well, they saw a very similar thing in the CAR T cell therapy patients. Not the lupus patients, by the way. In 2016 was the first FDA-approved drugs, CAR T cell therapy drugs for people who have B cell cancers. People with B cell cancers, they have a lot more B cells than people with lupus. And that's what a cancer is, a lot of too much of one type of cell. And so the CAR T cells go into the patients and attack all these B cells. And then just remember, every time that a CAR T cell and a B cell come together, not only does that T cell destroy the B cell, but it tells that CAR T cell to multiply and make more of itself. So since there's so many more B cells in people with cancer, you're getting a ton more CAR T cells being produced as well. You're getting all these extra cytokines being produced, and that's when you get this reactivation, the cytokine reactivation syndrome, where the immune system then attacks the organs, Fortunately, there's therapies for this. We use a medicine called Actemra or tocilizumab. It works very, very well. And in even people with B-cell cancers who get cytokine reactivation, the vast majority do very well with therapy. But occasionally, it can cause death. And so we have to worry about that as a possibility in lupus patients. But so far, we're really not seeing it. No severe cytokine reactivation syndrome occurred in any of those patients. CAR T-cell therapy can also cause severe neurological problems. We don't know why it happens, but it does happen in some patients with B-cell cancers. Fortunately, we have not seen any severe neurological problems in any of the patients who have been treated so far. Now, I do need to point out one particular type of CAR T-cell therapy for lupus patients. Novartis, it reported the results on 3 patients at our meeting that received something called YTB323. 2 of the 3 patients did actually have cytokine reactivation syndrome. Fortunately, it was mild in both of them, and they both did very well with Actemra therapy. So at least that's reassuring, but I don't know if I would want to use the Novartis product in my lupus patients if it's going to do that, but we'll have to wait to see the end of their clinical trials and see exactly what happened as far as the effectiveness and the potential side effects. CAR T-cell therapy has been used in other autoimmune diseases. The German group has used it in patients with other diseases like scleroderma. They reported their results in 3 scleroderma patients. Scleroderma is an autoimmune disease related to lupus where it attacks the skin and the blood vessels. Sclero means hardening of, derma means skin, and their skin can become very hard where they can't even hardly move their fingers, for example. But it does the same thing in the blood vessels, which can decrease blood supply to the organs. And they can develop severe lung damage and severe kidney damage. And unfortunately, it has a high mortality rate. And also, there's no FDA-approved drugs to treat the systemic problems from scleroderma. But the 3 patients who were treated with CAR T-cell therapy, the CD19 type, they all were doing fantastic at the 6-month mark. There were 3 patients with severe myositis. Myositis is an autoimmune disease that attacks the muscle. Myo means muscle, itis means inflammation of, and they had a particular type called antisynthetase syndrome. Well, at the 6-month mark and 1-year mark, 2 patients were in complete remission. 1 patient that they reported on was close to remission, but not there yet, but it was too early to tell. And by the way, antisynthetase syndrome is very hard to treat. They oftentimes fail multiple therapies. And then myasthenia gravis. Myasthenia gravis is another autoimmune disease that attacks the muscles. It can also be deadly because it can affect the muscles that help us swallow and breathe, resulting in death. Well, the type of CAR T-cell therapy called Descartes-08, that's directed towards BCMA, so it kills the plasma cells and not the other types of B cells. It was used in 14 myasthenia gravis patients, and all 14 of them did well. First off, the good thing about Descartes-08 is that chemotherapy is not needed. So that's a real good advantage compared to the other CAR T-cell therapies that we've been talking about. Also, it doesn't cause the cytokine reactivation syndrome, nor does it cause severe neurological problems either. All of the 14 myasthenia gravis patients got better, which is just phenomenal. But a problem with this type of therapy is that when you give this particular CAR T-cell therapy, the CAR T cells don't multiply in the body. They actually start to decrease over time. So at some point, the B cells do start to become active again. And they started to see myasthenia gravis antibodies return at the 9-month mark. The myasthenia gravis was still under very good control, but the antibodies started to come back. And so they think that patients with autoimmune diseases will probably have to get recurrent infusions of Descartes-08 over time. How well does it work in lupus patients? We don't know, but they are doing a clinical trial right now for Descartes-08 in lupus patients. Now, how about other side effects that occurred in autoimmune disease patients? Well, the myositis group And these patients were treated with the same CD19 CAR T cell therapy by the same German group that did the lupus patients I reported earlier. In the patients who had myositis, they did have some side effects. All three of them actually developed cytokine reactivation syndrome. Fortunately, all three did respond very well to Actemra. And then two of the three patients did develop a neurological problem called ataxia, where they had balance problems. It was mild. It went away on its own after a week. But this tells me that there must be something different about severe myositis that possibly or probably increases their risk of side effects to at least this particular type of CAR T cell therapy that's directed towards CD19. So it's not 100% safe in everybody. Other potential long-term side effects. B cells, again, are important for producing antibodies that protect us from foreign invaders. Another word for antibody is globulin or gamma globulins. And when we get rid of all those B cells with CAR T cell therapy, you could potentially end up with not enough B cells and therefore not enough antibodies. We call that hypogammaglobulinemia. And hypogammaglobulinemia increases the risk of infections. We're not seeing that yet with the lupus patients, but it's something we have to think about in the long run. And then, how about cancer? In November 2023, the FDA put a black box warning on the FDA-approved CAR T cell therapies that are available in the United States. And they stated that we have become aware of the risk of T cell malignancies with serious outcomes, including hospitalization and death, following treatment with BCMA and CD19 directed genetically modified autologous T cell immunotherapies. To put it in layman's terms or easy to understand language, the bottom line is that they had become aware that some patients treated with CAR T cell therapy against BCMA or CD19 actually resulted in CAR T cell lymphomas. Fortunately, it was not very many, but let's first talk about how this could have happened in the first place. So just remember, when you get CAR T cell therapy, we bring you in and we give you low doses of chemotherapy that gets rid of most of your T cells. And then we give you back the CAR T cells, and then the CAR T cells, when they start to interact with the B cells of your body and destroy them, they start to replicate and multiply throughout your entire body, and they go into the tissues of your body. But just think about it. What if one of those CAR T cells becomes rogue and starts to multiply and replicate and just multiply itself over and over again? This is the definition of a cancer. And so this can actually be a CAR T lymphoma or cancer of a lymphocyte. And CAR T cells are a type of lymphocyte, and that's what they've seen in a few patients. It's incredibly rare. Over 30,000 patients have been treated with CAR T cell therapy for B cell cancers so far, and only a few patients have been seen with CAR T cell lymphoma. But it's been seen in each of the FDA-approved products. And so therefore, the FDA has stated that this is most likely a class effect in these particular types of CAR T cell therapies. So we have to ask ourselves, is CAR T cell therapy worth it in patients with autoimmune diseases? This is very different than treating B cell cancers. The patients who have B cell cancers, they're going to die without treatment. So it's a life-saving therapy in many of those patients. But our patients are expected to live a long lifetime. Most of our patients will live a long lifespan. And do we want to subject them to possible side effects that we're not seeing yet? Could they get side effects like infections in the long run? Could they get the CAR T cell lymphomas like we've seen in some of the CAR T and some of the B cell cancer patients? And then there's other unknown side effects that might occur 10, 20, 30 years after treatment, and these are all big question marks. Also, should CAR T cell therapies be restricted to those types of therapies that are unlikely to cause CAR T cell lymphoma? The CAR T-cell lymphomas only occur in those CAR T-cell therapies where the CAR T-cells multiply over and over again in the body. But some CAR T-cell therapies don't do that. Remember I showed you in the earlier slides that the lentivirus inserts a DNA into the patient's T-cell and makes it a CAR T-cell. And then the DNA makes the CAR T-cell replicate over and over again. But Descartes-08, which recognizes BCMA, has RNA in it instead of DNA. RNA is ribonucleic acid. And when these CAR T cells go into the patient, they do not replicate over and over again. They start to disappear over time, so they don't become CAR T lymphomas inside of patients. And so these might be safer in patients with autoimmune diseases. The downside of it, though, is that this particular type of CAR T-cell therapy does have to be repeated again, maybe once a year, maybe twice a year. We do not know yet, and the lupus studies are just now starting. So watching this, I made this sound miraculous. It sounds like a cure, 100% of patients going to remission. That sounds crazy. None of them with severe side effects. I mean, that's just phenomenal. We don't have any therapies like that today. So you're asking yourself, are you a candidate for CAR T-cell therapy? And I hope that some patients will call up and see if they are possibly candidates. You do need to make sure you have severe systemic lupus, first of all. Most of these clinical trials, they do require a SLEDAI score of 8 or higher. If you want to learn how to figure out your own SLEDAI score, just go to my website, lupusencyclopedia.com, and then type in SLEDAI in the search engine, and it will take you to my blog article where you can figure it out on your own even. You also have to have failed multiple standard therapies. You have had to have failed cyclophosphamide and mycophenolate and maybe other medications. If those are true, then you can go to my website, lupusencyclopedia.com. If you type CAR T in the search engine, you'll easily find my blog article, or go directly there at lupusencyclopedia.com/CAR-T-cell-therapy-4-lupus. And then at the bottom of the page, you'll see the list of the clinical trials that are recruiting patients, and it even has the contact information. So you can contact them, or your doctor can contact them. Now, what is the future of CAR T-cell therapy for lupus? We really don't know until we have larger studies. Are the larger studies going to replicate what we've seen with the smaller studies? Are we going to see 100% remission rates that last for a long time? We don't know. And then the question is, how long do the remissions last? Do they last for 8, 10 years? The lifetime of the patient, we will have to wait and see. And are there side effects that could potentially be occurring over time that we're just not seeing yet? And so those patients will have to be studied for a long time. And then which patients should actually get which type of CAR T-cell therapy? Remember, the CAR T-cell therapy for CD19 didn't get rid of the anti-SSA antibody B cells. So should that particular type of patient need a different type of CAR T-cell therapy? And we're getting to the point where we may be able to identify nuances as far as what the immune system looks like in different lupus patients, and then give them a CAR T-cell therapy that's tailored towards their particular overactive lupus. And that is definitely a possibility. Then how about other autoimmune diseases? Will it work in Sjogren's disease? There is a clinical trial going on in Sjogren's disease, by the way. How about rheumatoid arthritis as well? And then should CAR T-cell therapy be reserved for only the sickest patients? Just think about that. Do I want to give a patient with mild lupus who's in remission on hydroxychloroquine a therapy that might possibly give them a CAR T lymphoma? And the answer is no for me, because that patient is most likely going to live a long, normal lifespan with just hydroxychloroquine, which is very safe in most patients. However, it's interesting to think about this. Because think of a 20-year-old patient who develops lupus, and if I get that patient into remission with hydroxychloroquine, and then that patient live for 80 more years, think about how expensive that is during the entire patient's lifetime. How many doctors that patient has had to see, how many co-pays for their doctor visits and their medicine and their laboratory tests, for example, and then give them just one CAR T-cell therapy and then it go completely away possibly. It might be worth the cost and the expense, especially if it's proven to be safe. We don't know yet. Well, I told you about the Asian woman from Germany who was treated in 2021. She wasn't the first patient treated with CAR T-cell therapy, and very few people are talking about this, by the way. It was actually the Chinese. A Chinese group treated a couple of patients in early 2019, and now have treated 13 patients with severe systemic lupus, and they presented their data at the American College of Rheumatology meeting. It has been over 3.5 years, and those two Chinese patients, are still in remission. Drug-free remission, taking no medications and with no major side effects. So I can't wait to see what the future holds for CAR T-cell therapy. They're going to have to keep studying these medicines over the lifetime of these patients, see how long the remissions last, see if we have any side effects. But isn't this exciting? Could this be a new chapter for the treatment of lupus patients? You know, today I have to see my patients frequently, at least every 3 months, doing laboratory tests, monitoring their medications. And just imagine if you could go into remission, be on no medications whatsoever, and see less of me as your rheumatologist, and live a much better quality of life, and not have to worry so much about your lupus. I certainly hope that that's the future for treating lupus patients. And thank you very much.
Thank you, Dr. Thomas, for describing the potential of these amazing new therapies. We hope you found this information as inspiring as we do, and hope that you will share it with others in the lupus community. Once again, a special thanks to our sponsors, Tin Rocket, Blue Eyed Crow Studio, 7 Minute Cinemas, and Pharma for their generous support. And we want to thank you for watching. We encourage you to visit our website, kaleidoscopefightinglupus.org, and follow our social media platforms for the latest lupus news, program information, award-winning blog articles, and so much more. Thanks for spending time with us. And remember, you are not alone. I'm Annika Hazlitt for Kaleidoscope Fighting Lupus. Please take care.